So I'm interested in how we turn genes on and off, and particularly how we turn genes on during the process of the formation of the brain. So this is a process we call neuronal development, and in this study, what we did was we focused on a gene called brain-derived neurotrophic factor, or BDNF for short. And we focused on this gene because it is very important during this process of forming a brain. And we wanted to understand what factors are important in making sure it's turned on at the right time and in the right location. And one factor that's very important in this is so-called enhancer elements. So an enhancer element is a sequence of DNA which is outside of a gene, but whose activation boosts the expression of the gene. And so we set out to identify regions like this for the BDNF gene. And we found an enhancer element, which is located along the chromosome from the BDNF gene. And when we activate this region, we can boost the expression of the BDNF gene. And conversely, if we inhibit this region, so if we prevent its activation, we actually prevent the BDNF gene being turned on properly. And not only does this prevent the gene being turned on properly, but this has really important consequences for brain development. So what we showed was that if we downregulate this enhancer, the neurons don't grow as well, and so you don't get this beautiful neuronal shape with lots of long branches, and also that when we actually look in the brain, the neurons aren't reaching the correct location. So one really important aspect of how enhancers actually turn on genes or boost their activation is something called DNA looping. And we often imagine chromosomes or DNA as a linear sequence. And so that will be as sort of depicted on this piece of paper, where you have the gene is a series of DNA bases up here, and the enhancer would be a series of DNA bases down here in green. But in the nucleus, within the cell, DNA is never stretched out like that. It loops, it bends, and it's moving all the time. It's very dynamic. And it moves with relation to different parts of the nucleus. And this is very important in how we turn genes on and off. And it's very important in how enhancers work. And what we indeed found for the BDNF gene was that as we go through the process of creating mature neurons, and when we switch on the BDNF gene, what we get is this loop forming between the enhancer and BDNF, so that we get a loop, a DNA loop of the intervening sequence. And at the same time, the whole structure moves away from the edge of the nucleus. And we believe that these factors are really important in actually enabling us to turn on the gene properly. We hope that this research will have quite far-reaching implications because of the importance of this brain-derived neurotrophic factor. So the gene that I've been talking about, BDNF, when it's turned on, it produces a signaling molecule, which is really important in brain development, as I mentioned already. But it's also very important in forming connections and developing neurons in adulthood. And these are processes that are important when we're learning and forming memories. So these two processes of development, when we're actually forming the brain and forming the neural network, but then also refining the neural network and changing it later in life. These are processes we're really interested in. And so understanding the regulation of a gene like BDNF is critically important, I think, in understanding the basic biology. But in addition, BDNF is known to be downregulated, so we see reduced levels in a variety of neurological disorders. So this includes disorders like depression, and also neurodegenerative diseases such as Alzheimer's disease and Huntington's disease. And so we know that the BDNF signaling is reduced in these disorders, but we don't necessarily know why. And so understanding these mechanisms by which we turn genes on and off and by which we alter the levels could be very important in understanding disease mechanisms. But also they provide a potential way to boost the expression of BDNF, which would likely have positive effects on these diseases. And so by potentially targeting the enhancer or targeting these loops, we may be able to find new ways in which we can upregulate BDNF signaling in the clinic.